This unit is going to cover what is called simple and differential staining. There is a thing called a simple stain, and there's also differentials referring to, there's different kinds, mostly a gram stain. So here's why you do this. One of the first things that a microbiologist does, if they find a microbe and they want to look at it, you're going to stain the cells so you can see what is called the cell shape slash arrangement. Now, under your study diagrams tab, it says here, a bacteria cell arrangement guide. And I like this. It's a PDF because you can make it big like that. You can blow it up and you can put it something, like this. All right, let me do this real quickly. This is supposed to represent, here's the nucleoid, and then you have the cell wall, cell membrane, DNA. Bacteria divide by a very simple process. It's called binary fission. And you can see one cell, that chromosome, one chromosome would be duplicated, and then it would split into two cells and two daughter cells. Very, very simple. E. coli, Escherichia coli, they divide every 20 minutes. Staphylococcus aureus or Staph aureus divides every 30 minutes. And some divide slower than that, but they divide very quickly. And what happens is, depending on the species, once these cells divide, sometimes they'll stay together, okay, after they divide. In groups, in pairs, it depends on the species. And that's what you have over here. And the first thing you'd want to do is you would want to determine what is the cell shape, and then you'd try to do what's called the arrangements. Now, this one kind of goes really detailed with the cell shapes, but it's a good diagram to look at. Go up here. Well, there are three basic shapes, and I wanted to have this while I up just to show you. There we go. So it has short rod. So basically you have rod shaped and then you have circle shaped. And I know some of these Coccobacillus, we're not really going to deal with that much, but here's Coccus or Cocci, circle shaped. Bacillus are rod shaped. This is a Spirillum. This is club, narrow on one end, swollen on the other. This is called spirochete, ribbon shaped. Vibrio, they're comma shaped. And then down here on the second part of that are the arrangements. If you start with Cocci, and I'll go through this with our diagram too, a diplococci is found in pairs. That means when you stain a slide, you look at it under the microscope, you'd see a whole bunch of pairs. And this is supposed to be a slide you'd be looking at. And you can see here, these don't look like pairs, they're more like in groups. And it's showing here's Cocci, but you can see they're all kind of clustered together. Tetrads are groups of 4. Sarcinae are groups of 8, and they stay together. It depends on the species. A streptococci, and you've heard of that. Streptococcus, strep throat. That's an actual genus of a bacteria, but in this case, we're talking about what's called the arrangement. And so this is a streptococci. Here is a staphylococci, which is a grape-like cluster. And that's what you would do. You would look at, you would try to see, with a simple stain, you can tell cell shapes and the arrangements. So I'm going to go to our outline. Common method for identifying different types of bacteria is using what is called the cell shape arrangement. And I just showed you this. They divide by a process called binary fission. Very simple. Here are the definitions. So you start with the shapes first. Here's Cocci or Coccus, circle shaped. Bacillus refers to rod shaped. And then Spirillum are S-shaped. And they're not usually found in groups. They're by themselves. They're called singles. They're by themselves. So here's some bacilli, there's some Cocci, and there's some Spirilliums. Now, as far as cell arrangements go, and when you're going to do an unknown report, you're going to have, the first thing you're going to do is you got to write in what is the cell shape arrangement of your organism. Diplococcus, 2 paired cells, and I have a diagram for that. Streptococcus, I showed you that. There's a chain of cells. Staphylococcus, grape-like cluster of cells. Tetrad, groups of 4. Sarcinae, groups of 8. Diplobacillus, 2 rod-shaped cells. So if we go back, and I think I have it pulled up still. I didn't show you those. Okay, there's Diplobacilli, 2 rods. V-shaped, don't see that very much. Streptobacilli is this. There's one called Palisades, which is sort of like that. It's uneven stacking of rod-shaped cells, but it's an arrangement. And there's the Palisades, or Pelicides. Clubbing, I showed you that one. Those are usually found in singles. One end is swollen, one is thicker. Spirillum are S-shaped. Spirochete are ribbon-shaped. So here's some Cocobacillus, supposed to be between rod and a circle. But here's a bacillus, a Diplobacilli. There's Palisades. Here's Streptobacilli. Coccus, Diplococcus, Staphylococcus, Tetrad sarsenate, Streptococcus. So when you do a stain, the first thing you're going to look at is what's the cell shape and arrangement. And you can tell that with a simple stain. Now, the procedure, and this is going to be here in just a second, there's a video of a person making a simple stain. You get a glass slide. You draw a circle on there. See this? That gives you the area for your target. You're then going to put a drop of water in that circle. You will then get some culture, either from a petri dish, or you can get one from a slant, or even a broth tube. So you're going to get some microbes, and you're going to move those microbes within the circle within the water. Tiny amount. And you do aseptic technique. You're going to heat the tubes. You're going to sterilize your loop. You want to make it as aseptic as possible. And like it says here, you're going to rub the inoculating loop in the water, and you would spread it out. Okay, the exciting part. You would let it air dry. And then you would have to heat fix the cells to the slide. And by doing that, excuse me, you would actually take this slide, once it has dried, and there's a video to show you this. Once it's dried, air dried, takes 10 or 15 minutes, you would then take the slide and you gently pass it through the flame in the Bunsen burner. And that does 2 things. It actually will kill the cells right here. Not only kills the bacteria cells, but it adheres them to the slide so they're not washed off during the staining process. It's a very important step. And if you're doing a simple stain, you can either do crystal violet, which is a purple stain, or saffronin, or even something called methylene blue, which is a blue stain. You would then flood the area with the stain. You'd let it sit. This here says, I believe it's 3 minutes. Let it sit for 3 minutes. You would then rinse it off or dip it in water to wash off the excess, and then you would dry it, blot it with this thing's called bibulous paper. These little books of paper you can blot, and now you have a simple stain. And you can put it under the microscope, and you can look and see what are the cell shapes and the arrangements. Okay, how to do a simple stain. A simple stain, all you can tell is the morphology, the cell shapes and the arrangements. And you can use different kinds of dye. This video he's talking about methylene blue or crystal violet or saffronin. So what you would do is you would make a circle, and this is how you know to work. You're going to put your bacteria there. You would then turn the slide over, and then he sterilized his loop, and now he's putting... What we do is we put water first, okay? And then we actually add the bacteria to the water. And I'm kind of watching this as you guys are. And that's what so there's a streak for isolation, looks like. Staph aureus. See the Staph aureus? Now he's mixing the bacteria into the water. Whoops. Okay, and we will have to pick that up with the gram stain. Now, a gram stain. This technique came from a guy named Hans Christian Gram. You can see him there, 1883. And a gram stain will tell you cell shape arrangement, but it also will tell you what is called, is your slide is your microbe gram positive or gram negative. You can't tell that with a simple stain. Simple stains, you can only tell the cell shape and the arrangement of the cells. And the process is this. Now, there is it's a 4-step process, and you have to do it. You would prep the slide just like that. You would get a blank slide, you draw a circle, You would put a drop of water there. You would then take some culture, some microbes, and smear it in the drop of water. You would let it air dry. Then you would heat fix it. Okay, right here. You would then pass it to the flame, kills the cells, adheres the cells to the slide. And then instead of just like in a simple stain, you would just flood it with one stain and leave it for 3 minutes. You do 4 steps. You do a primary stain, which is crystal violet, which is purple. So you would flood the area with that. You would wait for 1 minute. Man, I don't have that on here. I will add that to your outline. You do 1 minute. You leave it on there. Once you flood the area, you leave it on there for 1 minute. Iodine is the next step. Iodine is called a mordant. Iodine will actually bind with the crystal violet inside the cell. So when you do this first step, you're going to flood the slide with purple dye, and it's going to stain all the cells purple. Crystal violet would then attach to, iodine actually binds to crystal violet, and it keeps the stain inside the cell. It's called a mordant. It's an intensifier. So that's step number 2. Same thing. You leave this on here for 1 minute. And I will post this after here. All right. That's step number 2. Step number 3, you're going to add what is called a decolorizer, which is acetone alcohol. You put a few drops on there, and this is the key step. You don't want to overdo it because you can overcolorize your slide. You don't want to do that. But what will happen is the drops coming off will start running clear, and then you stop. And this is the key step. If you have a gram-positive, the acetone alcohol does not affect the cell wall, whereas if you have a gram-negative, it disrupts the cell wall and washes out the crystal violet. So the cells are no longer purple. And the last step, so this is not a minute or 30 seconds, this is just a few seconds. The last step of a gram stain, you add safranin. And you keep safranin on there for 30 seconds. And it's called a counter stain. And what will happen is if you have a gram-positive bacteria, and I'll explain what that means in a minute, gram-positives are going to retain the primary stain. It has to do with how their cell wall is built. Whereas a gram-negative will be decolorized and will take up the counter stain, and they will be red cells. And now you've put your bacteria into either two categories. They're either gram-positive or gram-negative. And that's very important. Not all cells can be gram-stained, but it's a way of determining what kind of cell you have, and it's a very simple process. I'm going to, hopefully this isn't going to kick me. Okay, yeah. All right. So, hopefully the breadcrumbs will work on this. Okay, good. So here's kind of where this really plays a good role. So this is a gram-positive bacteria. This is a gram-negative. And this is a pretty detailed diagram. The one I gave in your outline is a little easier to follow. See gram-positive? See this big, thick layer here? This is the cell wall. And it's made of something called peptidoglycan. What's interesting about that is that's penicillin's target. The reason penicillin was called the magic bullet and was such a great drug, antibiotic discovered, it only attacks bacteria cells because human cells do not have peptidoglycan. And it will bind in here and prevent a new cell from forming a cell wall, and that's how it kills it. So here's a cell membrane. Here are the phospholipids within that cell membrane. Go over that in lecture. So microorganisms that have this kind of cell wall, they end up staining purple during the gram stain process because the acetone alcohol does not disrupt the cell wall. This is what a gram-negative cell wall looks like. This is called a lipopolysaccharide. Now, lipids will dissolve in alcohol, an acetone. This is peptidoglycan. Little thin layer here, thicker layer here. So this is still the cell wall. It just kind of has 2 layers to it. And the thing about gram staining, and down here's a cell membrane. The thing about gram staining is when you add acetone alcohol to bacteria that are under that to bacteria that have a cell wall like this, the acetone destroys this top layer. And so what ends up happening during gram staining is you start out with crystal violet, which is purple, you add iodine, and then the decolorizer part will wash out all the purple because it destroys this outer layer because it's made of lipids and some sugar. Well, and starch, polysaccharide. And then what happens is that last step when you add a counter stain, the red goes inside the cell, and gram negatives will stain red color, whereas gram positives, they stain a purple blue color. It's a very simple process, and that way you can easily put something into categories right away. So this is a good diagram also. So here is crystal violet, here is iodine, here's the alcohol part, and there's safranin. And it gives you the whole procedure right there. Gram positives stain purple because of the cell wall. Gram negatives will accept the counter stain and end up staining, in this case, they look pink, but they're supposed to be kind of a red color. All right, let's go through a little simulation. I'm going to click on this. And by the way, you need a Flash plugin. That's actually on the course overview page. If you don't have one of those yet. But I'm going to click on that. And oops, hope I hit it right. Okay. These are really cool. All right, we're going to start. Let's do an example of a gram positive. What's going to happen during the gram staining process. All right, here's a gram positive cell wall. This is that thick layer. Don't worry about these parts coming off, but this is a thick layer of peptidoglycan. So what we're going to do at the bottom, we're going to add crystal violet. See, there's the C's. You click on it. When you stain the slide, you see how the little particles go inside, and don't worry about positive and negative charges. So now, during the first step of a gram stain, you have now stained the cells purple. That's what on the inside. Next step, you're going to add iodine. Remember? See, when iodine goes over the cell wall through the cell membrane, watch, it's going to come down here, and if you click on this, see it's binding to crystal violet. So now you got these larger molecules, so they can't leave the cell. All right, that's why iodine is called a mordant. It's supposed to intensify the stain for the cell. Now we're doing a gram positive example. And here's the thing, if you're wondering, well, why do you have to do it if you know it's gram positive? You don't know till you get to the end of the procedure whether you have a gram positive or negative. Going to add the decolorizer. Here's acetone alcohol. Now look what happens. It's evaporating the peptidoglycan, but it's not breaking it up. In fact, it's actually sealing it off. It's making it even more impenetrable. What a word. Okay, and then the last step here, look, see, the crystal violet cannot escape. All right? Because of the cell wall is so thick, the thick peptidoglycan. Now, when you add safranin, see, watch, we're going to add it. See, it's not able to get across the membrane. Okay, and then when you see it actually stops here because this layer and the cell membrane are not disrupted. And it says here to look at them under a microscope. So there we go. There's the gram positive, what you would see. So we're going to go back to the main menu. Still gram negative. So I'm going to select this one. So here is the cell membrane. Here's the thin layer, peptidoglycan, and here are the thick layers, what's called the lipopolysaccharide. So we're going to add crystal violet. Same thing, it's going to pass all the way across. It has to do with the charges. It actually pulls it inside of the cell. We're going to add iodine. Remember, iodine is called a mordant, an intensifier. So I'm going to click on this next. So then you have these big molecules of crystal violet, iodine here. Now we're going to add the decolorizer. Now watch what happens to the top up here when we do this. Boom. You see how it completely took it away? Now it's evaporating this thin layer of peptidoglycan. And now, because of that, see, the crystal violet is actually washed out. It's decolorized. And then when you add the last step, which is safranin for 30 seconds, boom, it goes under and accepts the counter stain, and now you have some red cells. So that's the whole thing. I'm going to go back to our diagram. So I'll post this with the times. This acetone is just a few drops. Yeah, it has to do the cell wall, gram positives, gram negatives, and see here's that thick layer of peptidoglycan. It is not disrupted by the acetone step. And here's the outer membrane. I like this one better. It's a little simpler. But see the cell wall's got the two layers. That layer is destroyed. This layer is thinned out. And then during a gram staining procedure, the decolorizing, this is decolorized, so that's why it accepts the counter stain. That's why they stain red and why these stain purple. Now, I have students before ask, why do you have to do this if you know beforehand? You don't know. You put your bacteria through those 4 stages, and at the end of it, if you have purple cells, you have a gram positive. If you have red cells, you have a gram negative. A couple of things that can cause a false positive. If your culture's too old, they have to be 24 hours, newly dividing cells. If your smear is too thick, it can't be decolorized. If you over or under decolorize, you don't want to leave it on there too long or do it too little. Or if you leave the counter stain on there too long. You're only supposed to leave it 30 seconds. If you leave it there for an hour, gram positives will eventually stain red because that dye will eventually work its way into the cell. Here's the procedure. I'll let you read through that. It goes through it step by step, and it even tells you, I know I put those times in there. There's 30 seconds. Here it is. So crystal violet, 1 minute, rinse, and you would actually, you don't blot dry between the steps. You're going to add crystal violet for 1 minute. You would then rinse or dab it in water. You would add iodine for 1 minute. You would then rinse it off. You add the decolorizer. You would rinse that off with water. Flood with saffron. You would actually rinse that as well, but then you're going to blot it dry. That's the entire gram stain procedure. We're going to talk about a gram stain now. Now, Biorad's a pretty reliable company.
In this video, you will learn how to perform gram stain.
Sorry. And it looks like they got them all lined up here. This is definitely acetone. That's saffron, and this is iodine, and that's crystal violet. And then you have some blank slides. You have a cultured disc there, and he went pretty quickly. And I was doing a little commentary on the simple stain. The part that was left off was you let the water and bacteria air dry, and then you heat fix it, and then you stain it. So this person drew their circle. They're now it looks like, I guess this is a loop, but they're now sterilizing their loop, aseptic technique. Now that's interesting. We do it this way. We can put a drop on the slide. That's a good way to do it. That way you're not getting very much water. If you put too much water in this process, it takes forever because you have to let the whole slide air dry. So what he's doing, or she, or whoever, it's a male voice. When you listen to it, maybe it's a guy, but so we got to sterilize the loop. So you put water on the slide. Next, and I'm watching this as you guys are, you're going to get some culture. Here, there's a petri dish and a little bitty tiny micro petri dish, and it looks like there's been a streak for isolation. So he is now going to spread that out. Now what we do is we spread out the whole slide, make it super big, because you do have to let it air dry. So sterilize the loop again, because you do have to, aseptic technique. Here we go. Dramatic pause. Now, what has happened, this is air dried. All right, takes 10 minutes, 15 minutes, depending on where it is. Now, the next step, once again, I'm watching this just like you, should heat fix the slide. Yep. Heat fixing does two things. It kills the bacteria cells, and it adheres the cells to the slide, so they don't slide off when you start staining the cells. You don't want them to come off the slide. So, heated that up, and this is what we would do. We would turn off our Bunsen burners, and now we're going to do the gram stain. So first player up, crystal violet. We have little droppers, but just like that, and man, they're really making it very clean. You have to have water during this process, so you leave it on there a minute. Oh, interesting. So he's dumping it in there, and you can see, look, when he takes it out, you can see that there were some cells stuck to the slide, and they're purple. Okay, grams iodine. You're going to add this for a minute. Iodine is called a mordant. It's supposed to intensify the stain inside the cell. It actually binds with the crystal violet and gets where you can't wash it out. So he's going to put this part in water as well, it looks like. We rinse, but that's fine. And you can see on the slide up there, you can see that little smudge in the middle. That's the cells that have adhered to the slide. Now they're stained, and they're purple because crystal violet was the start. Now, next you do acetone alcohol, and you only do a few drops. It's called a decolorizer. And this is the critical part. If you have a gram positive, it's not going to be decolorized. If you have a gram negative, it will be decolorized. And they did a lot of drops. We don't usually do that many drops, but you can see it got decolorized. So I'm guessing here this might be a gram negative. You never know, because it looks like it all came off as far as the crystal violet part. And the last step, for 30 seconds, you're going to add safranin, which is a red stain. And same thing. They're going to now dip this in water. They're going to leave it there for 30 seconds. And they're going to dip it in water. It's interesting he's doing all three, it looks like. I don't know if you'd want to do the one that has the acetone in it, but that's fine. But it looks like, to me, you can see the smudge on there. This would probably be a gram negative. Yep, and it looks like it would be. And what you would do now is look at it under a microscope, and you would look for the cell shape and the arrangements. Okay, last part of this unit, spore stains. A spore stain is, okay, first of all, there are some bacteria that are called spore formers. Bacillus, the genus Bacillus, the ones that you hear about, anthrax and all the bad things, botulism, those are spore formers. What happens is when a cell is under duress, like this one, some species, not all, can form what is called an endospore. Endo means within. And what happens is, you can see here it has DNA, it's got like a spore coat, and essential enzymes, parts of the cell, all go inside of the spore. Before the cells die, they form a spore, and I need to add to that an endospore around the DNA and the essential parts of the cell. And it's a dormant structure. Spores have sat for thousands of years, and people have inhaled them, and you start growing, and if it's a pathogenic thing, they can get sick. But here's an endospore, and there's different categories. This one's right in the middle. This is a terminal spore. This is a subterminal here. This is a central spore. So the species can vary between the species as to where it's located. Here's the actual spore, and it's showing that when conditions are more favorable, like for instance, if a cell had run out of water, oxygen, it could form a spore until the conditions improve, and it's showing it basically breaking out of a spore and growing again. Spores are not reproductive. They're just dormant structures. And here's an actual slide. These terminal spores would be found in the ends like that. These are central spores, and these are called subterminal spores. They're found between the middle and the center part. Well, a subterminal is between the end and the middle portion. Okay, so know these locations. Central spores in the center. Subterminal is not quite in the end and not quite in the center, and then a terminal spore is at the end. But there is a staining method that involves heat. It involves something called malachite green and then safranin. We're not going to worry about that, but the heat is required to get the stain inside of a spore. They're very resistant. They're hard to kill. You don't really kill spore formers really with liquids or anything. You kind of wipe them off. Autoclaving kills them. But anyway, that's an endospore.